And so we are going to welcome our next speaker, who is Gledis da Conceição. I hope I haven't made a mistake on the pronunciation, who is working for the National Administration of Conservation Areas. But Anna is going to be, did her master's. She's a recent master's graduate at Gorongosa National Park. And she's going to be talking about the relationship between morphology and echolocation parameters of bats in Gorongosa National Park.
Hello everyone. It is with great pleasure that I introduce myself to everyone who is connected to this Women in Bat conference. My name is Gledis da Conceição and I'm from Mozambique. I'm going to present my master research with the topic was the relationship between the morphology of the vestibular system and the echolocation parameters of bats of Gorongosa National Park, Sofala, Mozambique. I've been supervised by Aramonajan and Piotr Naskarek, two amazing people and bat lovers. Bats play an important role as seed dispersers and pollinators, and they also can suppress insect pest population, ensuring the effectiveness of agriculture production and maintaining the sustainability of ecosystems. They also are food for other organisms when we talk about ecological integration. Traditionally, bats have been identified based on morphometric features such as the size and shape of the skull and teeth, as well as the baculum, and more recently incorporating genetic and acoustic characteristics, allowing the identification of bats through an integrative taxonomy. Bats, the mammalian order Chiroptera, are divided into suborders, pteropodiforms and vespertiliforms. The pteropodiforms include all fruit bats of the family Pteropodidae, with some insectivore families that were previously included in a separated suborder, which is the Microchiroptera. The vespertiliforms includes the remaining microbats, and these bats produce echolocation signals in the larynx. In contrast, most species within the family Pteropodidae do not echolocate, except rhesutic Aegyptius that produce a primitive form of echolocation by tongue clicks. According with their echolocation types, high diet cycle echolocation and mid-long duration, narrowband calls denominated by a single constant frequency, while light diet cycle bats emit narrowband or broadband sound pulses separated by interpulse intervals that are much longer than the duration of the emitted pulses. And these species therefore separate the emitted pulse from the returning echo in time. Here we have some examples of bat families and echolocation types. For example, bats with quasi-constant frequency call are open-air foragers, which can catch prey in flight, use overlap sensitive or multiple harmonic frequencies modulated, and with the short duration at low intensities, and that one are the Mollucidae. And here we have an example of Cariphon pumilus. The frequency modulated are clutter edge foragers with low to intermediate wing loadings, such as the Naterius tebaca that here presented from the family Nateridae. For constant frequency are the ones that are clutter foragers, such as the Rhinolophidae, and Pteropodidae does not echolocate. Well, echolocation is a form of sensory behavior in which bats emit sound pulses, and based on the reflected echoes are able to form images of their surrounding in their brain. So this is associated with the auditory scene. The auditory scene applies widely to all hearing vertebrates, and all vertebrates that hear have an inner ear structure composed of a vestibular system, a labyrinth that is responsible for communicating with the external environment. Terrestrial mammals that are sensitive to loud frequencies, such as elephants and humans, tend to have a large ratio of the basal and apical cochlear turns, while mammals with relatively high frequency thresholds, such as rats and mice, have a lower ratio. The aim of this study was to correlating the morphological characteristics of the inner ear of a diverse fauna of bats in Gorongosa National Park with their echolocation parameters. Davis et al. In 2013 compared bats to non-flying mammals to determine the evolutionary consequences of echolocation and flight on the form of morphological of semicircular canal in bats using high-resolution microcomputed tomography. These studies expand on this approach. So, in this study will support the study carried by Davis and will allow a new method to be adapted within the integrative taxonomy for bat identification. I had two hypotheses. The first one, that theropodiforms have a different vestibular organ and bone structures than vestibular-uniform, which the prediction was theropodiforms have a higher number of turns in the cochlea. For the second hypothesis, there is a correlation between the structure of the vestibular organ of bats and the parameters of echolocation, where the prediction is if bats exhibit variation in the structure of the vestibular bone, this will be reflected in the echolocation strategy, and thus different forms of foraging. I used samples captured between 2014 to 2020 that were preserved in L. Wilson Laboratory for this study. So, in a year of 38 bat species were analyzed from 6 families from the theropodiforms and 6 from vestibular-uniforms. The stage of my research were skull extracted from the bat, and then I prepared a solution of a kale wedge with 20 milliliters of distilled water with 3 pills of kale wedge at 96%. And then I took to the microwave oven for a 30 seconds for rapid dissolution, followed by distracted the skull manually, and then I clean the skull manually, and extract the vestibular organs. I used a combination of focus stacked, we used the Helicom remote and Helicom focus, and I photographed it in each position multiple times, up to 30 times, at different focal distance to obtain focal stacked image with the imaging depth of field. And I took the photo of microscopic measuring scale, and then I prepared a folder. Then I tried to standardize the measurements. So to do that, I created 3D template because this one have different formats, and I also created the template in the Adobe Photoshop, and I did the measurement using ImageJPro. For a classification of bat frequency, were based on the taxonomy book of Monaghan et al. And for classification of the bat frequency, I used the echolocation category, which, for example, for bats that have quasi-frequency modulated call with high daddy cycle, I call it quasi-high. And for quasi-frequency modulated with low daddy cycle, I call it quasi-low. And from frequency modulated with low daddy cycle, I call it frequency low successively. To determine the relation of the number of cochlear curvature between the suborder, vestibular-uniforms, and theropodiforms, this test was performed to generate a bar graph. And for the second hypothesis, to determine the relation between the echolocation parameters and the vestibular system shape in 38 bat species, principal component analysis was done using PC1 and PC2, and all the analysis were made in air study. For intersubordinate variation in the number of tani in the bat vestibular system, it found that pteropodiforms have high number of cochlear spiral compared with vespertilineforms. So Davis et al., who analyzed a feature of 56 species, found significant difference in the Rhinolophidae and vespertilineform. Subsequent to their divergence, consistent with the multiple region hypothesis, he found that the two main groups of echolocating bats displayed different channel morphology in terms of size and shape in relation to bat mass and cochlear size, thus suggesting independent pathways and offering tentative evolutionary support for multiple acquisition of echolocation. For the second hypothesis, the relation between the vestibular system and echolocation, here we can clearly see, for analyzing for bats that have constant height represented for Rhinolophidae and Rhinonycteridae that have constant height are very distinct with the bats from which have quasi-low echolocation category represented from mob, condylor, and chirophone. Also, we can see difference here from the Embryonidae family with, for example, the Myonycteridae angolensis and Epomorphic wildback, the ones that are that have no echolocation. They are very distinct. I brought here some examples of bats with constant height that show similarity. For example, Rhinoloph Mozambique and Hypsidocafer have the structure a little bit similar. And also I brought the example of a researched Egyptian that have constant low, which show this structure. And this group don't necessarily need to echolocate to forage, so the bone would not have a complex. But because these lives in caves have this strategy of echolocation. Also, I brought here example of bats that have frequency low echolocation category that I didn't see similarity on morphological characteristics. And this I talk about Miniopteridae, Miniopterus wilson and Myotis tricolor. I didn't see clear separation between frequency low, represented for Miniopteridae, Vespertilionidae, and Nycteridae, and constant high, represented with Hypsidoridae, Rhinolophidae, and Rhinonycteridae, and quasi-height. So with this study, I was able to confirm that yes, there are differences in the morphology of the vestibular system. We could see how the bone have structure that between the species were different using different echolocation parameters. I could see that bats with quasi-low and constant high echolocation category, they have a great difference in their morphological characteristics, as they have different echolocation characteristics that are related to very different foraging strategies. So with this study demonstrated the existence of such relation and has found difference in the shape of the vestibular system, which can be of use in the integrative taxonomy of bats. So I recommend the use of sophisticated methodology microcomputed tomography, combining with this study. As you can see here, my study was through the session, and also would be good if the study was repeated between interspecies variation and increasing more families, because for this study I used the one that the O. Wilson Laboratory provided. And yeah, I would like to thank everyone that contributed for this study, and also would like all to thank for all people that organizing this event. Thank you so much.
Great, thank you very much, Anna. So we only have time for one very quick question. So yeah, really impressive work that you're doing, and I'm sure everybody will be very jealous with you working in Gorongosa National Park, and lots of people would love to do that. So Alex said, very impressive work, Anna, thank you. What do you think are some of the biggest challenges of the work that you've experienced doing that work in Mozambique?
Well, good morning, everyone. Thank you, Rachael. Thank you also, Alex, to make this question. Actually, this could be easy to answer, but at the same time, it's difficult because I would like to say when I started to work with bats, first, I'm not the one that I would say I loved bats since I was young. I started to love bats when I met them. I started to work them because I was one of the person that was scary of bats, but that's another conversation. Then when I started to work with bats, for me, it was think that it's like to hug them because they are cute, you know. And then I started to realize that there are many questions to answer related with bats. And when I started to make this question, in one of the examples is that from the master thesis, I wanted to count the bats in Mozambique, but that's not possible. Like when you think like you want to count the bats. So the biggest challenge is when I started this work, and likely I'm a lucky person because I have people that show me the direction. Example, Piotr Naskarek and Ara Monajan, and then because I wanted to know like what make the echolocation, for example, what make bats have echolocate, and then have what is related, is there any structure? But I didn't know how to do that because I didn't have the right methods to start to work with that. And then I realized that I can dissect and see what is have the same what we do with baculum. And then I work with them, but to dissect was not easy. I destroyed so many samples there, and this for me that was the biggest challenge. Also because I worked in Gorongosa, and there it's a conservation area which have like danger animals, let me say, and then to go to the field, it's not easy. You have to be like prepared and go with a big team, and then have the rangers to take care of the danger that you can find in the field. But also the biggest challenge that I still have for now, it's, well, I believe there are people that works in bats in different parts here in Mozambique, but we need to join our efforts to work together and see. For example, I asked the question to Dina about this colony, and then I realized that there's someone that started work with this colony, but why not we have? So this is my biggest challenge. Join the efforts to work together to have the battle, and then I don't want to be the only person that works in bats or think that I'm the one while have another people, because Mozambique needs first to have a catalog for Mozambique bat fauna. So this is my biggest challenge. Thank you, Alex, and everyone.
Yeah, thank you very much, Anna. So yeah, I definitely agree with you. I think there's a big challenge for bat researchers and conservationists across Africa. Yeah, so I just wanted to thank our last three speakers. I'm very impressive and inspiring. I'm sure you'll all agree. And it's just wonderful to see the work that's being carried out across Africa as well, and particularly by our early career professionals. It's quite exciting to see more researchers coming up.